I'm Becky Rosner, and I'm chair of the Department of Chemistry and Biochemistry, and it's my pleasure today to introduce our speaker. Our speaker is Dr. Erin Anderson from the Illinois Wesleyan class of 2007. Erin was a very accomplished student when she was with us here at Illinois Wesleyan. In addition to excelling in all of her coursework and being active in Greek life and being a great citizen in our department, she spent 3 of her years doing some research in the laboratory of Dr. Ram Mohan, where she was developing environmentally friendly catalysts for organic chemistry reactions. That outstanding foundation led to her going on to the Scripps Research Institute in California to earn her Ph.D., also in organic chemistry, and following that, she did postdoctoral work at the National Cancer Institute, where she worked on using infrared light to find diagnostic cancer innovations as well as cancer treatments. Most recently, she is at Loxo Oncology in Boulder, Colorado, where she's back to doing organic chemistry for drug discovery, working again on cancer treatments. She has been well recognized for this work, beginning as a student here when she received many undergraduate awards, including the Iota Sigma Pi Award for undergraduate excellence in chemistry. Iota Sigma Pi is a women's chemistry honorary, and they give only one such award a year nationally. Well, this success has continued. Erin is a National Institute of Health Fellows Award winner for research excellence, a finalist in the Chemical Biology Laboratory Original Proposal Contest. She has won the Center for Cancer Research Directors Innovation Award, and she has been the beneficiary of a Novartis Fellowship in Organic Chemistry for women and minorities. And I picked about a third of the things that are listed here. It's no surprise then that at least part of the reason that Erin is back this weekend for homecoming, even though it's not one of her even anniversary years, is that she is being awarded the Montgomery Young Alumnus Award. So with that, please welcome
Erin. Thank you all for coming. In my class, I'm going to talk about cancer. If anytime anything's not clear or you have a question, you can just raise your hand or shout out or however. Any interruption is fine. So today I'm going to talk about what cancer is. Then I'm going to talk about the current ways that we treat cancer. Then obstacles, why haven't we cured this disease yet? And then I'm going to talk about 2 areas of research that I worked in, my postdoc work, finding a way to use light to image and treat cancer, and then also what I do now, which is trying to design molecules for genomically defined cancers. So in your body, cells aren't supposed to live forever. Most of the time when a cell gets old, it will die, but sometimes a cell can undergo mutations, and then you have a cancer cell, and this cancer cell can undergo uncontrolled division and you get a tumor. And so cancer is a relatively young field. In 1975 is when Tharmas and Bishop first discovered the first oncogene. And now, just 45 years later, we can sequence tumor cell genomes in days. So we are making a lot of progress, even though we're not quite there yet. And so cancer isn't just one disease, it's hundreds of types of diseases. And there's actually 522 different human genes that have been implicated in cancer. 132 of these have been proven to be what we call drivers of cancer, so they're actually responsible for the cancer keeping to grow. And of those, they can be divided into 2 classes. There's things called an oncogene, and there's things called a tumor suppressor. So to use like an analogy like a car, an oncogene would be like if a gas pedal got stuck down. So basically the cells are just being told divide, 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 but we don't want them to. And a tumor suppressor would be like if you had a car that had broken brakes, so nothing's telling the cells to stop dividing. And so both of these are ways that cancer can start to form in your body. And this is just a pathway, each one of these guys is connected, each little block here is connected to the other ones, and they all feed back to each other, and all of these, every blob on this picture has been implicated in cancer, so you can see how complicated of a disease it is. It's also a disease of aging, and so these mutations you get in your cells, they accumulate, and what you get is these populations of cells that start growing, and you can see this is when the diagnosis of the cancer happens. In this case, it happened around this stage, but this is actually, about 17 years after the cells have started mutating in your body. And so that's why it's really important to get early screening and all that type of stuff. And they'll mutate until they reach one of these. They're called the hallmarks of cancer. And basically, once they get the ability to do one of these things, then the tumor is kind of off to the races, because any one of these factors can cause a tumor to start growing uncontrollably. And so now we know what cancer is. I'm going to go over the current ways that doctors prescribe to treat cancer. So the most common are chemotherapy, then there's radiation, immunotherapy, and targeted therapy are two newer treatments that are starting to be used more. So I call it cytotoxic therapy. It's chemotherapy, but it's, people have horrible side effects, and that's because it's a pretty unspecific therapy. It's just going after cells that are rapidly dividing. So there's multiple ways the drug might go after that. It might try to mess with the building blocks so the cells can't divide. It might break up the machinery so the cells can't divide. It does a lot of that kind of stuff. Unfortunately, cancer cells aren't the only cells in your body that are trying to grow and divide, and so that's why when people take chemotherapy, they have these other side effects where the drugs are acting on your normal cells, and so you can get hair loss, diarrhea, vomiting, nausea, stuff like that. And that's why people don't like taking these drugs. They have a lot of side effects, and they're really, we really need to find something better. There are some ways to target chemotherapy, so to take a toxic chemotherapy drug but deliver it only to the tumor. That's some of the research I worked on with the light stuff, so I'll go into that a little bit more later. But the idea here is basically that you take your cytotoxic drug and you package it in a way so it's not active, and then you put something around it, usually an antibody, that recognizes the tumor. And so then only when these guys recognize the surface of the tumor cell and bind, and then that's taken up, is the drug going to be released inside the tumor. And this is also done in a way, these things called prodrugs, where you have something attached to the drug that makes it not active, and then only when it's taken up into the cancer cell is it active. The challenge here is we are only just now starting to figure out all these markers that are on cancer cells, and you have to find the right markers that are just on cancer cells and not on your normal cells, or else we're going to get side effects again. So these are really important areas of research here. So in addition to chemotherapy, a doctor might prescribe radiation. And radiation, a beam of radiation is directed at the tumor. It can be directed externally, or it can be, a source of radiation can be implanted close to the tumor to deliver the radiation like that. There's also now different ways that depending on which type of cancer you have, you might take liquid radioactive element to treat the cancer. And this therapy, it's usually less side effects than chemotherapy, but there are still side effects when normal cells are affected. Much like chemotherapy, there's a way to target radiation, and with that you have called a bispecific antibody, so just an antibody that can bind two things. And what it does is you first put that in, it binds to the tumor cells, then you wait for the rest of it to clear from the blood, and then you'll put a radioactive element that binds that antibody in there, and that goes through and is cleared everywhere but where it binds to the tumor. And so that's a more specific way to do that type of treatment. Now, this is something that's getting more popular, immunotherapy, and a lot of work on this is done at the National Cancer Institute, so I saw a lot of this when I worked there. And what immunotherapy is when doctors, they take out a set of the patient's immune cells, and then they teach those immune cells how to recognize better the cancer, and then they put those immune cells back in, and your own immune system is treating your cancer. And this is a really powerful technique. It just depends on if your type of cancer is one that will respond to that type of therapy. But it's had some great results so far. And the last type of therapy I'm going to talk about is what I work on now, and it's my favorite type, it's targeted therapy. And it's what we do at Loxo Oncology. And in targeted therapy, we're still using small molecules, but instead of just going after any type of quickly dividing cell, we are trying to find the exact protein that's going wrong, the one that's acting as the driver, the one that mutated and has gone off, and then we're only going to treat that protein. And so there's not going to be a lot of side effects because that protein is often one that should have been shut down after development, and so it's not really something you need in your everyday life, so shutting it down doesn't give side effects. The key to this is that we have to do a lot of research to find out which are the drivers of cancer and treat the right proteins. And with targeted therapy, another caveat is that we have to genetically test the patients and then make sure that the patients have that mutation that's going to work with our therapy. And so that's the current treatments we have. So I'm going to go over obstacles in finding a cure, why it's so difficult. Yeah, exactly. And so this is such a big one that, yeah, when we say people have cancer, everyone that has cancer basically has a different disease. And not only that, even inside one tumor, the cells are different from one another. And so even inside a tumor, you could have different genetic mutations that are causing it to grow. We also don't fully understand how cancer works, so that's obviously a big one. And then another fourth reason is that even when a patient responds really well to a treatment, we can have the patient will stop responding and the cancer will keep growing. So the cancer can find a way to evade the treatments, and you can get an evolved resistance to treatment. And then the final is just a medicinal chemistry problem. Sometimes proteins aren't druggable. So now I'll go into each one of these in more detail. So this is a very complicated slide, but we don't need to read the words. Across the top is just types of cancer. So every vertical row is just a type of cancer. And across the vertical axis is just different mutational signatures. So this is a certain type of mutation, and you can see it's implicated in every type of cancer has some patients that have this type of mutation. If you look at it from the other side, you can see that if you have bladder cancer, it might be driven by any one of 5 major mutations. It's not just one disease. And so all of that just makes it so when you go and you get the cancer diagnosis, there's kind of a lot more research that needs to be done in order to figure out how to treat it and figuring out exactly what's causing the cancer. And this is like the targeted medicine, kind of. It's the same concept. It's just the term for the sequencing part of the targeted treatment. But precision medicine is basically, in a conventional approach, we take a bunch of patients that have breast cancer, we give them a drug, and it worked for some, it wouldn't work for others. And, we get these clinical responses in clinical trials that aren't that great. And so that might cause the drug not to be approved. It might, or on the converse, it also causes us to give chemotherapy drugs to a lot of people that maybe wouldn't benefit from them because we haven't tested their cancer to make sure they would. And so one thing that we advocate for at Loxo Oncology is that each patient, when you're diagnosed with cancer, you need to get some sequencing done to find out exactly what types of mutations are wrong in your cancer. And from there, it's much easier to divide patients in subsets with maybe a certain protein mutation. So maybe all these patients have breast cancer, but this patient has, a mutation in protein X, this mutation in protein B, protein C, and then you can give people a drug that's going to treat that, and you'll have much better responses. So that's really an important part of cancer treatment.
The numbers on the right are based on what?
The numbers on the right are like clinical responses. So basically when we go to clinical trials and we give the patients
Are
the
they
drug.
real numbers to date, or?
Oh, no. Well, this is just a slide I pulled from the internet. This isn't from my company or anything. Yeah. So the second thing, tumor, each tumor in and of itself is very different. So when you have a tumor, you'll have your original tumor cells, but then a lot of times they grow into, like, each color here is a different set of genetic mutations. And so within a tumor, you'll see a lot of different types of cells. And this is really alarming if you look at, this is a scan of a guy who had cancer, and they genetically tested all of his lesions, and every color here is a different genetic mutation is highly upregulated. And so you can see how when they give you a cancer treatment and maybe we didn't get it all, well, they're not. On the left is, I think, just a PET image, and then on the right, they're just coloring it based on the sequencing they did. So they're just adding color to the PET image. I think it's PET. I think, yeah. I think to me, yeah, and what's more is that even though these tumor cells are different and all different, what's even more complicating is they talk to each other and they help each other, and this is another thing that we're just now starting to understand. So in this, they have a tumor that has two different types of cells. When they take the first cell and put it in a mouse, they don't get tumor growth. When they take the second cells as well and put them in a mouse, they don't get tumor growth. When they put them both together, then you get this highly aggressive tumor. And so this is another thing that we need more research on, is how tumors cells talk to each other. And so we know they talk to each other, that's one complication, but there's a lot we don't know about tumors, and this is the tumor microenvironment. And so a tumor isn't just made of tumor cells, it also has blood vessels, it has immune cells in there, there's cells that make machinery for the tumor, all types of stuff. And so it's quite a little ecosystem, and there's a lot of research being done right now to figure out how this microenvironment affects how the cancer can grow. So now is the part we don't fully understand how cancer works. So the first part of this is a driver versus a passenger mutation. So if we think about like the car, driver is the one kind of deciding where the cancer is going to go, what it's going to do. The passenger is just along for the ride. And this is important because when it seems simple to be like, oh, we'll just take a bunch of tumors, we'll genetically sequence them, we'll see which mutations are going wrong, and we'll treat those mutations. But some of those mutations are the cause of the cancer, and some of those mutations are just there because you have a cancer cell that's really rapidly dividing and can't divide very well, and then it has a lot of mutations. And so this is really important for what I do, that we have to make sure that anything we're treating clinically is one of these driver alterations, because you don't want to put a lot of research in and not be treating the actual cause of disease. And then additionally, tumors shut down our immune system. And so tumors actually, they can send out these things, the actual term for them is called death ligands, and they can tell your immune cells to just stop. And so there's a lot of complications in figuring out how to get your immune cells to recognize cancer again. And now for evolved treatment resistance. So in these tumors, if we treat them with targeted therapy And we really are very active against the mutation we're trying to kill, and we kill all of those cells. This goes back to the fact that I told you there's a lot of other types of cells with different mutations. Well, some of these cells can persist, and then your cancer can come back, and when they genetically sequence those tumors that come back, you can see that reflected in the genetics of those tumors, that they came from the cells that weren't treated the first time. And so that's one thing we really look at is trying to figure out how all of these driver genes are talking to each other, how all these proteins are causing cancer to keep going. And the last one is just the medicinal chemistry problem. Sometimes it's just hard to find a small molecule that can bind to some of these proteins. And so this is what I do daily. I look at a protein, and then I look at how a small molecule, so the proteins we're trying to treat, they usually have a substrate, a natural substrate in the body that they bind. So we look at how that natural substrate binds, and then we try to find a small molecule that would bind like that. And so we're basically looking in these little type of pockets and trying to fit small molecules in there. Unfortunately, there are certain types of proteins that instead of having little pockets where they bind small molecules, whenever they talk to each other, it's just a protein and protein talking. And so these are these big flat surfaces coming together, and it's harder to find like a protein like this big and then a small molecule this big. So it's hard to find a small molecule that can bind and stop these two huge things from talking to each other. But one really awesome thing that just came out last month is Amgen actually just created the first small molecule that binds these undruggable proteins. So hopefully we won't be calling things non-druggable anymore because people are having progress finding molecules that can treat these. So this is exciting. So now I'm going to talk, the last two things are my research and how to treat cancer. The first is using light to image and treat cancer. And this is work I did at the National Cancer Institute at Fort Detrick in Frederick, Maryland. And so why do we want to use light to treat cancer? Well, if you're thinking of how you want to turn something on, light's a very easy thing to use because you can shine light where you want it and you can turn it on when you want it. So you have spatial and temporal control over whatever type of therapy you're doing. And then also, light is relatively non-toxic. That's why in terms of what type of light we use, we don't use UV light, which is toxic. We use IR light, near IR light, so right in the red region of this visible light spectrum. And another advantage that IR light has over UV light is there are less things in the body that IR light absorbs, and so it can penetrate much deeper into tissue. And you can see that with the laser pointer here. The green doesn't penetrate as far as the red here. And so light is already used in cancer. Light's used to image cancer, and it's also used to treat cancer currently. And so first I'll go over the way it's used to image cancer. So when I started working at the National Cancer Institute, one thing I learned is when surgeons are cutting out cancer, they're relying a lot on their expertise. It all kind of just looks like a pink mass area, and so the surgeon has to rely on their expertise to cut out all that they think is cancer, and then they give a sample of the margins to a pathologist. The pathologist will then say, we think you've got it all, or maybe you should cut more, and that's what they'll go based off that. It's with fluorophore. We give the patient a small molecule that binds to the cancer, and then you just shine a light, just a regular light, not invisible light. And when you have this, so you put a fluorophore, so you give the patient a fluorophore, which is just a small molecule that fluoresces, and we've designed these molecules so that they bind specifically to cancer, and so then when the surgeon shines light, he can see the fluorescence light up, and then you're just cutting between something like pink and, or green and not green, or colored and not colored, and that can help make that easier to get those margins and make sure you have all of the cancer there. And one thing that's kind of cool, an engineering project, and a lot of this is done in St. Louis, which is near where I'm from, so it's pretty cool. But engineers are working to incorporate all of these things together in one set of goggles, and so just the surgeon would be looking and they could see a field of vision that was the anatomy and a field of vision that was the dye.
Supposed to be a surgeon. All that stuff.
Yeah. Yeah. So how does this work? Well, basically, we're giving the patient a fluorophore, and this is the type of stuff I worked on, was designing which of these fluorescent molecules would bind to cancer specifically and then light up. And so when you hit the fluorophore with light, and usually it's a laser, but it's an IR laser, so it's not going to burn you or anything, not toxic form of light. So then that fluorophore will absorb a photon and be excited, and it will become what's called excited state fluorophore. And then at that point, that fluorophore can relax back down to the ground state, and then that's fluorescence, and that's what you see when you look at that. And the issue and why we were looking for better fluorophores is because as the fluorophore cycles through this process, it starts to degrade. And so as of now, if people would have a long-term surgery, they would have to be given another dose of the fluorophore. And so we tried to make fluorophores that were longer lived and then also more stable, so they would be easier to use in surgery. And then the other thing that I spent the majority of my postdoc working on was using IR light for treatment. And so IR light is currently used for something called photodynamic therapy. In photodynamic therapy, a patient is given what's called a photosensitizer, and that's just a non-toxic small molecule. And this photosensitizer then goes all over the body. We wait a bit of time until it localizes to the tumor. It can either be localized to the tumor through a targeting mechanism, or it can be localized to the tumor. I told you tumors have blood vessels. Their blood vessels grow really fast, and they don't grow very well, and so they're leaky. And so sometimes you can target a molecule just trying to get it to fall out of the tumor's leaky blood vessel. So after the photosensitizer is localized to the tumor, then we shine light into that area. The photosensitizer is then excited, like that fluorophore was, and it reacts with oxygen all around it. And the reactive oxygen species are actually what does damage to the cancer. And so this is now getting to be a pretty popular technique. When it was first created, it was first approved in the 60s, I think, and the photosensitizers weren't so specific, and so it's really only done in places without a lot of sun, like Russia or Minnesota. But now that we can localize these better, a lot of people are doing it all over. It's even used to cure acne at dermatology clinics. And so this same general scheme of therapy is what's used for all types of light therapy. In photoimmunotherapy, it's just the same process, only we're using an antibody to target it here. And then in light-triggered drug release, that's what I work on and what I'm going to go over next. And with that, we're just trying to do this step, and then when we shine light, we're not having this happen, but we're having a chemotherapy drug release right at that spot where we shine the light. And so the technique we use is called near IR uncaging, and it's just the caging group is just basically something on there that makes the drug not work. And so when you have these two things bound together, this drug isn't active. However, when you shine light on it, it breaks the bond between these two, and the drug is then active and able to do its job. And this makes it so you don't have that drug systemically all over your body. You just have it released at the site where you shine light. And so what our actual molecule looks like is this. The blue would be, this black would also be blue. It's all part of the caging compound. And then we put the drug on top here. And what we wanted to do is we wanted to make a molecule that was active both in that normal PDT, photodynamic therapy, like I told you, was approved a long time ago. And then we also wanted it to be active as a way to uncage the drug. So we wanted to make something that could do two different things. And the reason we wanted to do this is because, like I told you, since tumors are very different all over, some parts of the tumors have oxygen and some parts don't. And so having something that treats both of those things is important. These are just the questions we wanted to ask. Will it work? And so first we make the molecules, and chemistry is it's just like cooking. Basically, you just put these two things together and heat them up, and then you get this guy out. And so once we made the molecules, we put this drug molecule. This is called cumbravastatin, and it's a chemotherapy drug. It's the drug we wanted to see if we could release. And so this is a picture of these molecules when they were put into cells. And this is a stain for the nucleus here. This is a stain, or this is a fluorescence of our compound. So our molecule, on top of everything else it does, it's actually also fluorescent. So we see the fluorescence. So that's a nice way to check that your compound actually gets into cells, is we just look and it is in there. And so then we tested if this drug could kill cancer cells. And so this is just a test for if the cells are alive or not. So this is 100. If it's at 100, they're living, and the lower it is, the more cells die from treatment. And so this is when we treated cells with our compound in the dark. Our compound is not toxic in the dark, which is what we wanted, because we don't want it to be activated by light. However, when we shine light on the cells, we see cell death happening in a lot of the cases. In the solid bars here, it's when we have cells that have oxygen, and so we see moderate cell death in both cases. And this is, we think, if we go back to the two things I wanted to happen, so that would be this top arm, where you're getting, within oxygen conditions, you're getting photodynamic therapy effects to kill the cells. And then in these checkered bars, we incubated cells without oxygen. And what you see here is that when we put the active drug on cells without oxygen and shine light on it, we see a lot more cell death. And so what we thought that was from is that we were releasing our drug. And we can test this with a type of test called FACS. It's where you can sort what the cells look like. And we know that our drug should make cells stop in this part, if you remember, when we learned about mitosis and how cells divide, cells, if they get hit with this drug, they're stopped at this G2M phase. And this is just what the diagram looks like when you have the drug. You get a lot of this G2M. And then the same thing when we take our active drug and we irradiate it. Only when we shine light on it and the cells don't have oxygen do we see the same big uptake in these G2M cells here. And so that's just evidence that we are uncaging the drug and that this is working. And the other molecule doesn't do that. And then the final way we just proved that what we wanted to happen was working is this experiment called photopatterning. And in this, we put black masking tape over half of the cells. And so when we shine light on them, it's only the left-hand side of the well that's getting light, and the other half, it stays dark. And the important thing to remember about this experiment is when cells are, or when the molecule is killing cells via photodynamic therapy, it's actually exciting oxygen that's around it to kill the cells. And that oxygen that's excited, it's so reactive, it just reacts right where it is, and it can't really travel throughout the well. Whereas a small molecule gets released, say the drug gets released, it can go throughout the well, the cell well, and act on all the cells. And so what we expect is that you'll just see cell death on the left side when there's oxygen, but you'll see cell death on both sides when there's not. And that is in fact what we see. So here in this, living cells are stained this dark color, and then all the dead cells, there's no staining. And so you can see when we shine light on our molecule without oxygen, we get cell death all over the well. When we shine light on our molecule with oxygen, you just get cell death where the photodynamic therapy could happen. And so both ways that we're looking at this drug are working, and just the control showing that with the control molecule, we're not seeing that, which is what we expect. So then we put this into mice to see if it would work in vivo. For this, we chose pancreatic cancer. The reason we chose pancreatic cancer is because it's known that pancreatic tumors are highly heterogeneous. The red parts here, this is cells that don't have very much oxygen. The blue parts are cells that have oxygen, and then the white parts are actually parts of the tumor that have died. And so you can see how kind of what a mosaic this is, and how it'd be helpful to have a drug that can kill both the cells without oxygen and the cells with oxygen. And so with this, just like with the human, what we're going to do is we're going to inject our drug into the mouse, have this drug localized to the tumor. We can, like I told you, since our drug's fluorescent, we can then image and make sure that our drug got to the site that we want it at. We can then image, or then we can shine light and hopefully break apart that drug at the site of the tumor and get the tumor to shrink. And so this work is ongoing now. I had to leave, but what we have shown is that we need to formulate these. And so basically, this molecule doesn't have very good drug properties in and of itself. So we put it in these little fatty bubbles and then give it to the mice. And it has a lot better properties in the blood. And so that's where they are now, is working with this formulation and seeing if that works in vivo. So now, just the last few slides are the work I do now at Loxo Oncology, and we were just bought at the beginning of this year. We're now a wholly owned subsidiary of Eli Lilly. So,
Which company?
Eli Lilly.
Oh, Eli
bought us,
Lilly.
yeah. So, at Loxo Oncology, we work on targeted therapies. So, the first thing we want to do is we want to find out which are those driver mutations that are driving cancer. The next thing we want to do is we want to make sure that we design compounds that are super selective. So we only advance compounds into the clinic that are only treating certain proteins. We don't want a lot of side effects. And then probably the most important part is just making sure that the patients that we give these drugs have had genetic testing and can benefit from these drugs because they have the mutations that we're trying to treat. And so if there's anything we take from this talk today, it's just get genetic testing if you're ever get cancer. So these are the different types of mutations we work with, what is actually happening at the gene level. Sometimes things can click together that shouldn't have ever been together, and that can just cause the protein to go. There's also amplifications, overexpressions, all types. So we work on all of these types of mutations. And so what we do is this is like a picture of the protein is here in white, and then our molecule is here in gray. So basically, all day I look at these types of images and I try to find out, if you put a piece here, are you bumping up against the protein too much so it wouldn't fit? Stuff like that. And so the way we do this is based a lot on computation as well as structure. So basically we get the 3D picture of the protein, usually from an X-ray, and then we put that in the computer. Then we get these pictures out, and then we design a bunch of compounds. Then I make the compounds, then from the cells, see which ones are active, and go from there. And what this has allowed us to do is to make drugs that are super selective. So this is just one of those parts when I showed you that big chart of all the different proteins that can be activated. This is just like the top left of that, but this is all the proteins in that top left part. And the caveat to them is they all look very, very similar. So it can be hard to find a small molecule that's only going to bind to one. But what we do, like this is a certain drug here, and you can see this is why it has side effects. It hits so many different proteins. Whereas our drug, this was approved this year in November, our first drug, and it's just very selective, only hitting 2 proteins that are going wrong in a specific type of cancer. And so this is the drug, and these are, it's called a waterfall plot, and they usually never look this pretty. Our company is super happy to get one that looks like this because this is the change in tumor size, and every color is a different type of cancer. That's why we call, the way we do research is called tissue agnostic. So we're not necessarily looking for just people with breast cancer, we're looking for people with this. This is from our actual clinical trial. This is from our actual clinical trial. Yeah, we don't, it was just approved in November, so we don't have much data back from that yet, but we're definitely still gathering data and making sure.
Hopefully the same color appears more than once.
Yeah, so there are
A
some.
different trial?
They're the same trial, but it's a different patient. Different patient. And yeah, each individual bar here is actually an individual patient. And you can see, so this is like 100% of the tumor. So no tumor was seen anymore for like these patients. And then you can see varying degrees of change. But in the conventional approach, a lot of times you would only see like 30% across the board because you weren't selecting the patients. So this is actually pretty incredible that we're getting this much of a response. And in this chart, every time there's a red dot, that's a complete response. So there was no trace of the cancer anymore. Each one of these bars is also a patient. And every red dot is a complete response. So the cancer, they couldn't find a detectable part of the cancer. And you can see that this is one thing I'm proud of that our company does, is you can see that people do opt to stay on the drug even after their complete responses. And I think that's a testament to how we do a good job of making selective drugs that don't have a lot of side effects. So yeah, so that is our first drug, and hopefully we will have more. That's what I'm working now to do, is find more drugs like this. But yeah, another thing, kind of speaking to the clinical use of the drug, one thing we do is we get back all the data to make sure that any patients, how I said that sometimes they can get a resistance from therapy. We make sure to check if any of their tumors are mutating, so we can make a drug that would then treat that new cancer.
Can you comment on the affordability of these treatments for the patient?
I don't know. Sorry, I don't really.
That's a question.
No, it is a question, yes. And yeah, this particular drug was actually licensed by Bayer from us, so we don't have any control over that anymore. But yeah, it's a very real concern.
Most of them
for
are
sure.
very expensive, yeah.
Yeah.
My husband probably took one a day, or 6,000 for a month's supply. But his medical, the drug insurance fee, so you don't pay a hundred, but yeah, most of them are pretty expensive.
Yeah.
Turnaround time, you talk about, say, a recurrence, and you retest, genetics have changed.
Like how fast?
Patient only has a certain amount of time. What kind of turnaround time do you picture for you in the lab to create a cocktail?
Yeah, so it's really important that we have to start the second generation therapies as we start the first generation therapies. So they do tests in the lab to try to predict which mutations will happen, and then those are the ones we start on right away, because you're very right, we can't get one out. If we would just find out about a mutation, we can't get through the clinic fast enough.
For example, this drug, When,
For this drug, we already have a second generation
Yeah,
that
no.
we approved, actually.
when did groundwork start on this, do you think?
Oh, this is actually an interesting story. So this was actually a pain drug that was shelved and not worked on at another company, and then Loxo Oncology, my company in 2013, licensed this and started, so they were kind of starting from a little bump because a lot of the medicinal chemistry had already been done with it. But yeah, it was actually a drug that was being looked at for pain and was not working very well, and so it shelved for that. And then when they found this germline mutation, we did a screening and they licensed this drug, and then that's how they found that. So.
Do you have a history of artificial intelligence analyses for your results?
We don't yet. Now that we were bought by Eli Lilly, they say they have a site in San Diego where they're starting some of that, and so I think we're going to start some more of that. And I think that'll help a lot on like in this part, looking for just searching quickly through the targets, and then also I think it'll help with trying to predict mutations. So we don't do a lot of that yet, but I think it is coming.
Some of us are having an overwhelming response to all this. It's like sort of a preview of a 1 or 2 semester medical school class. But that's my question really. Where would this kind of presentation fit into a medical education for oncologists or for basic medical studies? Would this be early or second or third year, or how? At what point do they start really dealing with this kind of detail?
Yeah. So I definitely learned a little bit about it. You know, I learned about drug discovery in undergrad, like the process of it. But as far as a lot of the stuff I didn't learn until my postdoc, probably when I went to the National Cancer Institute and went through one of the presentations there, learning a lot about it.
But you're PhD, not MD, right?
Yeah, PhD. Yeah,
Well,
for a medical, yeah, I guess I don't know for.
As a retired doc, medical education has also changed. And so it's, you know, when I took it, you had first year basic textbook stuff, second year,
This kind of stuff?
a little bit, yeah.
I mean, it would
Well,
be
that
this-
didn't exist 50 years
It
ago.
would be this kind of stuff now.
We had the chemical rubber handbook. But so many curriculums now, they start patient contact on day one.
Yeah.
And as the problem and the patient present themselves, you learn the pharmacology. So really, it's going to be,
You learn it when you need it.
you're going to be exposed right away. And she is right. Biochemistry is going to prepare you, hopefully, to comprehend a
Yeah,
bunch of
yes,
this.
sure. Oh,
That's one of our big, like I know, like when we did have a sales force for this drug, they were just basically trying to educate doctors about genetic testing, and just that's the only thing we really push for is just trying to, because it's not just our drug. Like there's a lot of drugs that are targeted therapies, and if you can figure out which mutation is going wrong, then you can maybe give the patient a list of like actionable drugs they could use, and so that's like the biggest thing we push is just trying to get genetic testing out there, because it's definitely not something, like one of my coworkers, her mother-in-law got cancer and she had to fight the doctor to get testing, and said you don't need it, and she fought and got it, and she did have an actionable mutant actually, so it's definitely not as widespread as it should be.
And there's this whole world of armamentarium. Have you dealt with viruses at all as carriers
No,
for
not
treatment? And so that's being done. And but I think med students are going to be exposed very early, understand it way better than I do. I was just curious, you kept referring to small molecules, and I love your pictures because that's my chemistry. Draw a benzene ring and do stuff with it. And like I said, the chemical rubber handbook. Where does small molecule meet medium molecule? What kind of sizes do you?
I work with probably anything under 500 molecular weight. So yeah, pretty small. Like I think when people have like small peptides, they still call them small molecules, but yeah. Oh, adult. Atomic mass units.
The other thing you talk about mutations, that's why people have one cancer and sometimes get another cancer and another cancer years later. The PET scan helps a little bit to see if it's spread everywhere, but still.
Yeah, and it's interesting because we don't know necessarily when it's hard to tell sometimes when it metastasized, like has that metastasis been there for a while and it's just now growing, or did it just, all that stuff is, stuff we have to figure out, because it's,
Where are all these clinical trials being held at?
We do clinical trials all over the world, actually. And so there's cancer sites all throughout the U.S. where they'll do it, and then actually all throughout the world.
MD Anderson, Sloan.
Yeah, I think we do all those. But yeah, there's like a huge list of cancer sites, and actually, I think the people in my company, we've driven across states for different people and stuff. So yeah. So this is my conclusion, so it's good timing. This is just, all hope is not lost. There's a lot being done. We have all these obstacles to a cure, but genetic testing is getting better every day, and we're getting ways to figure out exactly, what type of cancer you have. There's immunotherapy. We are learning better about how the drivers work. We're doing a lot of research on every part of the tumor so we can understand that and how tumor cells talk to each other. We're also, working on second generation therapies, combination therapies, that we make sure we fully eradicate the cancer and improving medicinal chemistry every day. So that's my class.
And Erin, can you need to go to like a research hospital to do the kind of genetic testing that you need to be able to do this, or can most any hospital?
Yeah, I think most any hospital can do it. I know, like my coworker, I know they had to fight a little bit with insurance to get it approved because, I just know it was like she had to fight with her insurance for, or her mother-in-law's insurance for like a month. But then it's just something that's like sent out by her doctor. But yeah.
Yeah, it's drawn local and sent off to standard.
Medicare probably doesn't do the last thing. The tumors can be surprising. My husband, great robotic Italian guy at the University of Illinois, was sure, get it, it's a great spot, gets in there. I mean, he was almost crying when he talked to me, because when they got in, the tumor had, he said they make blood vessels. It had rerouted every vessel in the abdomen except the aorta around the tumor, and they were like, no one could operate it, because there was too much vascular around the tumor.
Yeah, I mean, it's,
And all the scans said it would go for it.
yeah, and when you get in there, it just looks totally
Couldn't go
different.
for it.
Yeah. So yeah, that's a very,
Are there any further questions? Let's thank Erin. There are probably a lot of science-philic people in this room. Just to let you know, biology and chemistry are hosting a tailgate at the south edge of the Shirk parking lot tomorrow before the football game. We welcome you all back.